Good day, class. I am James Cedric Mojica, and I'm your professor for today. And we will discuss the ultraviolet and visible molecular spectrometry. But first, let us have an overview for our lesson. And please observe the picture below and answer my questions. What do you think will happen to the person that is directly receiving ultraviolet light from the sun? Can anyone answer? Okay. And number two, knowing that ultraviolet light from the sun is harmful, what methods should be done to prevent its harmful effects? That's right, we will wear um, clothes that will protect our skin and use umbrellas. And now, I have here jumbled letters and please pasugatan po natin ito. Number 1, can someone please answer? Yes, it is ultraviolet. Can you please share your insights about ultraviolet? Okay, thanks. The next one. Yes, it is wavelength. So another, please someone provide their insights and understanding about wavelength. Okay, thank you. The next one is, yup, that is absorbance. Someone please provide their insights about absorbance. And for the last one, please answer this and provide your insight about the word. Yes, that is precip. These terms will be related for our topic. Now, let us proceed for our lesson proper. We will discuss about the ultraviolet and visible molecular spectrometry. It is a technique used to measure how molecules absorb ultraviolet and visible light. When a sample is exposed to light in these ranges, it absorbs certain wavelengths. And by analyzing the absorbed light, you can determine the concentration and identify the molecules in the sample based on the specific wavelength they absorb. This technique is commonly used in chemistry and biology to study the properties of various substances. In this lesson, we have key concepts. One is the electromagnetic spectrum, where light is a form of electromagnetic radiation. It exists in a wide range of wavelengths, from radio waves to gamma rays. The visible spectrum is a small portion of spectrum encompassing colors from violet to red. Next one is the absorption and emission. When a molecule absorbs light, it gains energy. This energy can be released as heat or by emitting light of a different wavelength. Next will be chromophores. Chromophores are certain functional groups in molecules, known as the chromophores, and they are responsible for absorbing UV or visible light. These groups contain conjugated double bonds or aromatic rings. In conjugation, the longer the conjugated system in a molecule, the more likely it is to absorb light at longer wavelengths. The presence of these groups can influence the absorption properties of chromophores. For example, electron donating groups tend to shift light absorption to longer wavelengths, while electrons with withdrawing groups shift it to shorter wavelengths. In the steric hindrance, the spatial arrangement of atoms and groups can affect the overlap of orbitals involved in conjugation, influencing the absorption properties. Let us now proceed to the quantitative application analysis of the topic. First is identifying unknown compounds. By comparing the visible spectrum of an unknown compound to the spectra of known compounds, it is often possible to identify the unknown. And in determining functional groups, the presence or absence of a certain functional groups can be inferred from the absorption bands in a UV visible spectrum. For example, consider benzene. It has a conjugated ring of 6 carbon atoms. Due to its conjugation, benzene absorbs UV light in the range of 250 to 260 nm. If we introduce a substituent group such as nitro group, the electron withdrawing effect of the nitro group shifts the absorption to a shorter wavelength. Now for the class questions. What are the main factors that influence the UV visible absorption of a molecule? Then, how can UV visible spectroscopy be used to distinguish between isomers of a molecule? And lastly, what are the limitations of UV visible spectrometry? Now, for your class activity, I have proposed some questions, and the first one to raise their hand will have the chance to answer. Number 1. What is UV visible spectrometry? Correct. And what are chromophores? Yes. And how does conjugation length affect the mass of molecule? For the next batch of questions, what is the effect of electron donating or electron withdrawing groups on UV visible absorption? Can UV visible spectrometry be used to distinguish between isomers of a molecule? And lastly, what are the limitations of UV visible spectrometry for qualitative analysis? Okay, since you already understand the first part of our lesson, let us now proceed for the quantitative application of the UV visible spectrometry, which is the Beer-Lambert law. The Beer-Lambert law states that the absorbance of a solution is directly proportional to the concentration of the absorbing species and the path length of the light through the solution. Mathematically, it is expressed as absorbance equals molar absorptivity times path length times concentration. So refer to this concept. A is the absorbance. Then that letter E is the molar absorptivity, or a constant characteristic of the absorbing species. B is for the path length of the light through the solution. C is for the concentration of the absorbing species. In the linear relationship of Beer-Lambert law, linear relationship between the absorbance and concentration is implied, and provided the solution is dilute and there are no interfering substances. The molar absorptivity is a measure of how strongly a substance absorbs light at a particular wavelength. It is a constant for a given substance and can be determined experimentally. Then, these are some concepts that is related to the quantitative analysis of Beer-Lambert law. One is determining concentration. If the molar absorptivity and path length are known, the concentration of an unknown solution can be calculated from its absorbance. Then in quantifying reactions, by monitoring the absorbance of a reactant or product over time, the rate of a chemical reaction can be determined. In analyzing mixtures, if the molar absorptivities of different components in a mixture are known, their concentrations can be determined simultaneously using a technique called multiple linear regression. The path length is the distance the light travels through the solution. It is typically measured in centimeters. Then, these are some concepts that is related to the quantitative analysis of Beer-Lambert law. One is determining concentration. If the molar absorptivity and path length are known, the concentration of an unknown solution can be calculated from its absorbance. Then in quantifying reactions, by monitoring the absorbance of a reactant or product over time, the rate of a chemical reaction can be determined. In analyzing mixtures, if the molar absorptivities of different components in a mixture are known, their concentrations can be determined simultaneously using a technique called multiple linear regression. For example, suppose you have a solution of a known concentration of a dye. By measuring its absorbance at a specific wavelength and knowing its molar absorptivity and path length, you can use Beer-Lambert law to calculate the concentration of the dye in an unknown solution. Since you already know the fundamentals and quantitative applications of UV visible spectrometry, I have here presented a video of how a spectrometer works.
Here's how a spectrophotometer works. A lamp provides the source of light. The beam of light strikes the diffraction grating, which works like a prism and separates the light into its component wavelengths. The grating is rotated so that only a specific wavelength of light reaches the exit slit. Then the light interacts with the sample. From this point, the detector measures the transmittance and absorbance of the sample. Transmittance refers to the amount of light that passes completely through the sample and strikes the detector. Absorbance is a measurement of light that is absorbed by the sample. The detector senses the light being transmitted through the sample and converts this information into a digital display.
So, a UV visible spectrometer is a laboratory experiment used to measure the absorbance of light by a sample as a function of wavelength, and it consists of several components. First is we will prepare standard solutions. We will prepare a series of standard solutions containing known concentration of the analyte. Then we will measure the absorbance of each standard solution of a wavelength where the analyte absorbs strongly. Then we will create a calibration curve, which is the plot the absorbance values against the corresponding concentrations of the standards, and this will create that calibration curve. And we will measure the absorbance of the unknown sample at the same wavelength. And lastly, we will determine the concentration by um, using calibration curve of the unknown sample based on its absorbance. So, questions for the class. What are the main factors that influence the UV visible absorption of a molecule? Someone please answer. Then, what are some common applications of the Lambert law in analytical chemistry? Now, here are some real-life applications of the UV visible spectrometry. In water quality monitoring, it is used to measure the concentration of pollutants in water, such as nitrates, phosphates, and heavy metals. By analyzing the absorbance of these substances at specific wavelengths, environmental scientists can assess the quality of water bodies and monitor pollution levels. Then, in drug quality control, it is essential for ensuring the quality and purity of pharmaceutical products. It is used to determine the concentration of active pharmaceutical ingredients in formulations, monitoring drug stability, and detect impurities. In quality control and authenticity, UV visible spectrometry is used to analyze the color, clarity, and authenticity of food and beverages. For example, it can be used to determine the concentration of pigments in fruit juices, detect adulteration in wine, and monitor the quality of dairy products. So, since you already know the principles of spectrometry, let us now have a quiz. Number 1, what are chromophores? A. Functional groups that absorb UV visible light. B. Atoms that emit light. Or letter C. Molecules that scatter light. Number 2, how does conjugation affect UV visible absorption? A. Longer conjugation shifts absorption to shorter wavelengths. Longer conjugation shifts absorption to longer wavelengths. Letter C. Conjugation has no effect on absorption. Number 3. What is the Beer-Lambert law used for? Letter A. Determining the concentration of a solution. Letter B. Measuring the wavelength of light. And letter C. Analyzing the structure of a molecule. Number 4. Which component of a spectrometer selects a specific wavelength of light? A. Monochromator, B. Detector, C. Light source. For the last number, what is the relationship between absorbance and concentration according to the Beer-Lambert law? Letter A, linear. Letter B, exponential. Letter C, inversely proportional. Okay, the quiz is done. We will have now an additional activity that you will answer at your home. So you will create a paragraph containing 5 sentences on how can UV visible spectrometry help you in your respective career as a pre-service teacher in science. So that's all for today, and goodbye class.